Hi everyone, welcome to Unique Biology. This is the continuation of cell organelles part 1. The remaining topic of cell organelles I have explained here in part 2. So let's start. Centrosomes are composed of centrioles, and centrosomes are organelles that serve as the main microtubule organizing centers for animal cells. Centrosomes, they are present near the nucleolus and present generally in animal cell, and these are absent in prokaryotes and higher plants. So these are generally they are present in the animal cell and they are absent in prokaryotes in higher plants. So these are generally they are present in the animal cell and they are absent in prokaryotes in higher plants. Centrosomes, they are made up of barrel-shaped clusters of microtubules called centrioles. Centrosomes are mainly involved in cell division. Function of centrosome. At the end of prophase of mitosis in animal cells, centriole duplicate and new daughter centrioles are formed. So at the end of the prophase during mitosis, these animal cell centriole they duplicate and new daughter centrioles are formed, and they give rise to basal bodies from which cilia and flagella arises, and the distal centriole of sperm give rise to axonomes or axial filament of sperm tail. Now next we will study about the lysosomes. Lysosomes are single membrane sac-like structure found in most of the eukaryotic cells but absent in prokaryotes. So these lysosomes, they are the single membrane sac-like structure and they are mainly found in eukaryotic cells but absent in prokaryotes. The membrane is lipoproteinaceous in nature and they are absent in RBCs, yeast and euglena, and cholesterol and corticosteroids give stability to membrane and they prevent damage by enzymes. The matrix of the lysosome contain hydrolytic and lipolytic enzymes, and lysosomes are regarded as suicidal bags. These lysosomes are regarded as suicidal bag as they take part in intracellular digestion, especially when the cell experience starved condition or acute problem of survival. Lysosomes show polymorphism and exist in 4 types. They are primary lysosome, secondary lysosome, tertiary lysosome, and autolysosome. This primary lysosome, they are formed by the Golgi complex and contain enzyme synthesized by rough endoplasmic reticulum. And secondary lysosome, they are formed from primary lysosomes, and these are also known as digestive vacuoles. And the tertiary lysosomes, the indigestible or undigested food present in secondary lysosome forms materials of tertiary lysosome, which is known as residual bodies. And the function of the autolysosomes, these differentiate from primary lysosome and are self-motivated to engulf the cellular contents, that is self-digestion or autolysis in the deficiency of food. So that's why they are known as the suicidal bags, as they take part in intracellular digestion. The structure of lysosome. This lysosome consists of lipid layer, hydrolytic enzyme mixture, glycosylated membrane, transport protein. So this lysosome, they have a simple structure. They are spheres made up of a lipid bilayer that encloses fluid that contains a variety of hydrolytic enzymes, and lysosomes are formed by budding of the Golgi apparatus, and the hydrolytic enzymes within them are formed in the endoplasmic reticulum. And a lysosome is a membrane bound cell organelle that contains digestive enzymes. Lysosomes are involved with various cell processes. They break down excess or worn out cell parts. They may be used to destroy invading viruses and bacteria. Function of lysosomes. Lysosomes break down old cell organelles to their respective molecular component for resynthesis of organelles, thus help in cell recycling. So these lysosomes, They break down the old cell organelles to their respective molecular component for the resynthesis of the organelles. And also they are involved in the destruction of cells of the tail in tadpole during metamorphosis to form an adult tadpole. Next we will study about the nucleus. The nucleus is a dynamic cell organelle which actively and randomly control the functioning of all other cell organelles. So this nucleus, it is a dynamic cell organelle which control the functioning of all other cell organelles, either electrochemically or neurochemically. And this nucleus, it is also known as brain of cell, as it helps in maintenance of DNA and RNA transcription, basically an eukaryotic cell, and control gene expression and mediate the DNA during the cell cycle. And generally there is a single nucleus in a cell, but some cells also contain 2 or more nuclei. So in a cell it is only a one single nucleus, but some of the cells contain 2 or more nuclei. We'll study about the structure of nucleus. The nucleus is composed of following components. Here you can see in the structure. It is composed of nuclear membrane, euchromatin, karyolymph, nuclear membrane, perinuclear space, chromocenter, chromatin, nucleolus, pore, and heterochromatin. First, the nuclear membrane. The nucleus is bounded by a double-layered nuclear membrane with pores at intervals, and the space between the two membranes is known as the perinuclear space. And this nucleus, it is also consists of nucleoplasm. Within the boundary of nuclear membrane, specialized cytoplasm is present, known as nucleoplasm. Specialized cytoplasm, it is present in the nuclear membrane, it is known as nucleoplasm. And nucleolus, the nucleolus is a spherical dark body seen in contact with a specific chromosome at a point called nuclear organizer. And chromosomes are the thread-like colored bodies that are in the intranuclear position, which act as the vehicle of hereditary and variation. And euchromatin is also present. Euchromatin refers to less coiled region of chromatin. It is less stained and contain genes which are active, whereas heterochromatin refers to the chromatin which is highly coiled. It takes more stain and contain genes which are less active or inactive. So this was about the structure of nucleus. Next, functions of nucleus. It is responsible for storing the cell's hereditary material or the DNA, and also it is responsible for coordinating many of the important cellular activities such as protein synthesis, cell division, growth, and a host of other important functions. Next, the ribosomes. Ribosomes are asymmetrical particles which are dense granular structure found attached to endoplasmic reticulum. These are the dense granular structure. They are mainly found attached to the endoplasmic reticulum and found floating freely in the cytoplasm. And these ribosomes, they are also present in mitochondrial matrix and stroma of chloroplast. They are the sites of protein synthesis. So these ribosomes, they are the sites of protein synthesis, and also they are present in both prokaryotes and eukaryotes. Chemically, they contain 40% of proteins and 60% of rRNA, and their number in a cell varies from 10,000 to millions. Function of ribosomes. These ribosomes are the sites of protein synthesis. Here, the protein synthesis takes place. Free ribosomes produce enzymes for intracellular use. Intracellular use such as skin cells or erythroblasts. And these bond ribosomes synthesize enzymes for extracellular use. Extracellular use such as pancreatic cells, plasma cell, etc. Next, we will study about the vacuole. These are non-cytoplasmic areas present inside the cytoplasm and bounded by a single layer of semi-permeable membrane called tonoplast. So these vacuoles, they are non-cytoplasmic area They are present in the cytoplasm and they are bounded by a layer of semipermeable membrane called tonoplast. They are bounded by a single layer called tonoplast. And vacuoles mainly occurs in eukaryotes. In animal cells, the size of the vacuole is smaller, whereas in the plant cell, the size is larger and it is central in position. Structure of vacuole. A vacuole stores salts, minerals, pigments, and proteins within the cell. The solution that fills a vacuole is known as the cell sap. And the vacuole is also filled with protons from the cytosol that helps in maintaining an acidic environment within the cell. A large number of lipids are also sorted within the vacuoles. Function of vacuoles. A vacuole is a membrane-bound cell organelle. In animal cells, vacuoles are generally small and help in sequester waste products, whereas in plant cells, vacuoles helps in maintaining water balance. Sometimes a single vacuole can take up most of the interior space of the plant cell. So in animal cell, it generally helps to sequester waste products, whereas in plant cell, it helps to maintain the